Welcome to Frederick National Lab Technical Services video series. Today we have Dr. Claire Deliage, who is the head of tissue analysis core in our AIDS and cancer virus program. Dr. Deliage, welcome and thank you for talking to us today about unique services your lab provides. Please tell us a little bit about services that you offer.
Yes, with pleasure. So the Tissue Analysis Core has an overall mission to provide state-of-the-art analysis capabilities, including immunofluorescence, single and double label immunochemistry, and in situ hybridization, and quantitative image analysis within the AIDS and cancer virus program, but also in support of intramural and extramural investigators. So we're offering multiple services, and the first one is a specific staining and quantification of the CD4 positive T cells within any type of tissue, utilizing masking on non-T cells that are expressing CD4. So this helps to address the number of potential target cells within the tissue and gives insight on the severity of the depletion of the cells post-infection. And also it can help assessing the effect of a treatment on the recovery of the cells within the tissues. We are also offering assessment of fibrosis in the tissue using specific immunochemistry detection and analysis for collagen 1 staining. This is a common pathogenicity associated with HIV and SIV infection. But the services or assays, I should say, that people are contacting us for the most for is our RNAscope assay. So it's based on an approach developed by the company ACD, but we adapted it. We adapted this really next generation in situ hybridization to the HIV field needs. We are offering the detection, localization, and quantification of cells harboring the viral RNA and or the viral DNA in any type of tissues. We can visualize the signals using chromogen approach or fluorescence approach. And we can even combine this detection of the viral genome with cellular markers. So these approaches really provide important and novel insights regarding the localization and the phenotypic features of reservoir cells within their resident native tissue and microenvironment, which is simply not achievable with any other approaches.
Thank you for that description. So what makes your services unique?
Yeah, that's a good question. So aside of our unique expertise and knowledge in tissue staining, one of the really unique strengths of our services offered here at the TAC is our distinctive way to quantify the data. So having beautiful pictures is a good thing, right? But being able to quantify, put a number on those information is really powerful to address the needs in any type of research now. So we're closely working actually with Dr. Yanling Liu, which is a scientist here at the ABCS at Frederick, to develop really unique tools using the deep learning approach to quantify each of our staining. We're actively working on the manuscript describing this new approach to detect and localize the to quantify each RNA and DNA scope signals and report those data to the number of CD4 T cells that are the target cells of the virus within any type of tissue, which has never been done before in the HIV field. So the TAC really offers full service, I will say. We are reporting to each of our collaborators representative pictures and quantification for any services provided. We're also offering a lot of flexibility and options in our staining to be able to really answer any specific questions that a principal investigator can have when designing any type of study. So we're capable to run our assays also, which I think is important to notice, on fresh tissue, but also on frozen and on fixed tissues, which is a really broad type of samples.
Perfect. So what type of research or researchers do you normally support?
Yeah, so we are supporting a wide range of research and researchers, actually. Our first goal, of course, is to make sure to support the needs of our program, the AIDS and Cancer Virus program. But we're also providing our expertise to external investigators, to private company or any university. So most of our collaborators work on studies relevant to key questions to the HIV/AIDS research. However, the TAC has been offered support to other type of research, like studies on gamma herpes viruses, on Zika virus, and lately we also been involved in SARS-CoV-2 studies since we have adapted our assays to different viruses.
Perfect. Dr. Deliage, thank you so much for talking to us today. And if you're interested in learning more about these services, please do not hesitate to contact us. Thank you.
Thank you.